Welcome back to the deep dive. Today, we are tackling something absolutely fundamental, DNA replication. It's really the engine room of life itself. And when you look at the sources, the whole process seems almost, well, impossible. It's this constant, incredibly fast race against, molecular chaos.
It really is. I mean, think about what life means. It fundamentally involves making accurate copies. If copying your genome had an error rate of, say, just 1 in a million base pairs, which sounds pretty good, right?
Yeah, 1 in a million seems tiny.
But it would mean introducing something like 6,400 mistakes every single time one of your cells divides. That's just completely unsustainable for complex life.
Okay, so if that's the chaotic version, what's the reality? What level of precision are we actually looking at?
The actual precision is just astounding. The sources often use E. coli. The bacterium is a great example. Its genome's about 4.6 million base pairs.
Right.
Now, if it copied itself at, let's say, 1,000 nucleotides per minute, which sounds reasonably fast, it would still take over 3 days for one round of replication.
3 days? But E. coli divides, what, every 20 minutes or so under good conditions?
Exactly. So the reality is way, way faster. The actual speed is closer to 1,000 nucleotides per second.
Wow.
And the accuracy, the fidelity, we're talking less than one mistake in a billion nucleotides copied. That combination of speed and accuracy is, well, it's what we're going to unpack today.
So our mission for this deep dive is to look at the sources, understand the mechanisms, the key proteins involved, and tackle that big existential crisis for our kind of chromosomes, the linear ones, the fact that they tend to get shorter every time a cell divides.
That's the plan. We'll trace how we figure this out, look at the molecular toolkit, and confront that end replication problem head on.
But before we dive into the how of replication, maybe just a quick pause on the why. Why DNA? I mean, before we knew the mechanism, why did scientists even suspect DNA was the hereditary material in the first place?
Yeah, good point. It really came down to circumstantial evidence, as the sources remind us. DNA was in the right place, the nucleus, associated with chromosomes. Its quantity behaved correctly. The amount of DNA precisely doubled during the S phase, the synthesis phase of the cell cycle, right before division.
Ah, right.
And if you compared diploid cells with two sets of chromosomes, they had exactly twice the DNA content of haploid cells, like sperm or eggs. It was really the only molecule in the cell that ticked all those boxes. It behaved like the blueprint.
Okay, so DNA looked like the stuff. Then Watson and Crick published the double helix structure in '53. And that structure, with its complementary strands, A pairing with T, G with C, it immediately suggested a copying mechanism.
It screamed template, didn't it? But how exactly those parent strands were used to make new molecules, that was still up for debate. It wasn't immediately obvious.
Right, there were a few competing ideas.
Exactly. 3 main theories were on the table at the time.
Can you walk us through those?
Sure. First, there's the one that turned out to be correct, the semi-conservative model. The idea here is the parent double helix unwinds, and each of those original strands serves as a template to build a new complementary strand.
So each new DNA molecule ends up being a hybrid, one old strand, one brand new strand.
Precisely. Then you have the conservative model. This proposed that the original parent molecule somehow stayed completely intact, both strands together, and directed the synthesis of an entirely new double helix made of purely new material.
So you'd end up with one molecule that's 100% old DNA and another that's 100% new DNA.
Correct. And the third option was kind of the messy one, dispersive. This suggested the original parent DNA got chopped up into pieces, and these pieces were scattered sort of randomly throughout both new DNA molecules.
So both strands in both new molecules would be a mix, a patchwork of old and new bits?
Yeah, a real mosaic.
Okay, 3 distinct possibilities,
Right.
and the way they figured out which one was right is just...
Yeah, it's one of the most beautiful experiments in biology, isn't it? The Mendelssohn-Stahl experiment.
Absolutely elegant.
Yeah.
Its genius was in using isotopes to track the DNA by weight, by density.
How did they set that up? They started by growing E. coli for generations in a growth medium containing a heavy isotope of nitrogen. Nitrogen is a key component of DNA bases, right? So this made all the bacteria's DNA heavy.
Okay, so all the initial DNA was dense.
Then they abruptly switched the bacteria to a medium containing only the normal lighter isotope. Any new DNA synthesized after that switch would be light.
And they could separate the heavy, light, and any intermediate DNA using centrifugation, spinning it really fast.
Exactly. They'd release the cells, extract the DNA, and spin it in a cesium chloride gradient. The DNA molecules migrate to a point where their density matches the density of the gradient, forming a band. Heavy DNA sinks lower, light DNA stays higher.
So the crucial moment, after one round of replication in the light medium, what did they see?
They saw only one band of DNA, and its position was exactly intermediate between the heavy and light positions.
Ah, so that single result immediately knocked out the conservative model, right?
Instantly. Because if it were conservative, you'd expect two distinct bands after one generation. One heavy band, the original parent molecule, and one light band, the completely new molecule. Finding only intermediate weight DNA meant every single DNA molecule had to contain some of the original heavy stuff.
Okay, so that leaves semi-conservative, where each molecule is half old, half new, or dispersive, where everything is a mix.
Right. The decider was the next round of replication. They let the bacteria divide again in the light medium. Now, what would each model predict?
Well, if it was dispersive, everything would still be mixed, maybe just getting progressively lighter overall, but still fundamentally intermediate, right? Maybe the band would shift up slightly.
Exactly. You'd expect maybe one broader band, still intermediate but lighter than before. But what Michelson and Stahl actually observed were two distinct bands. One band was still at that intermediate position,
The half-old, half-new molecules from the first generation.
and a second band appeared that was purely lightweight DNA.
Ah, and that pure light band could only form if entirely new light strands were being synthesized against the light strands made in the first generation. That confirms the semi-conservative model.
Precisely. The semi-conservative model perfectly predicted that pattern. Half intermediate, half light after two generations. The dispersive model couldn't explain the appearance of a purely light band. It was just definitive proof. Beautiful logic.
Okay, so semi-conservative it is. Each strand is a template. But actually making this happen requires a whole suite of molecular machines, right? A big protein complex.
Oh, absolutely. It's a real molecular factory. You need the basics, of course.
Yeah.
The single-stranded DNA template, once you unwind it, the building blocks, those are the deoxyribonucleoside triphosphates, DNTPs, DATP, DCDTP, DTTP, DTT.
The A's, T's, C's, and G's with their energy packs attached.
Right, and the main worker enzyme, the star of the show,
Yeah.
DNA-dependent DNA polymerase.
But DNA polymerase has that fundamental flaw, doesn't it? It's a great builder, but it can't start a new chain from scratch.
That's its Achilles heel. It can only add nucleotides onto a pre-existing 3' hydroxyl group, that specific chemical hook at the end of a nucleic acid chain. It needs something to extend.
Which is why we need a primer.
Exactly. And strangely enough, that primer isn't DNA, it's RNA. A short stretch of RNA, maybe 10, 12 nucleotides long, is synthesized by an enzyme called primus.
And primus can start from scratch.
Yes, because primus is actually an RNA polymerase.
Hmm.
And RNA polymerases, unlike DNA polymerases, don't need a primer. So primus lays down that little RNA starter strip, providing the necessary 3-OH end, and then DNA polymerase can take over. It's like the starting pistol for DNA synthesis.
Okay, let's picture this starting in bacteria, like E. coli. It all begins at a specific spot, the origin of replication, orosty. What kicks things off there?
It starts with initiator proteins like DNA and E. coli. These proteins bind to specific sequences within orosty, particularly an AT-rich region because AP pairs have only 2 hydrogen bonds, making them easier to pull apart than GC pairs.
So the initiator proteins start to melt the DNA open a little bit.
Exactly. And that little opening allows the main unwinding enzyme, DNA helicase, to bind. Helicase is the real zipper opener. It latches onto one strand, typically the template for the lagging strand, and uses ATP energy to just motor along, disrupting the hydrogen bonds and forcing the two strands apart, creating what we call the replication fork.
But as soon as those strands are separated, they're sticky, right? They'll want to snap back together or form internal hairpins.
Correct, which is why you immediately need SSB's single-strand binding proteins. They quickly coat the exposed single strands, keeping them extended and preventing them from re-annealing or getting damaged, like little protectors.
Okay, so we're unzipping. Helicase moves forward. SSBs coat the strands. But unzipping a tightly wound helix must create tension further down the line, right? Like overwinding a rope.
Massive torsional strain builds up ahead of the fork. If you didn't relieve it, the whole process would grind to a halt. That's the job of DNA gyrase, which is a type of topoisomerase found in bacteria.
And gyrase does something pretty radical, doesn't it?
It does. It actually makes transient double-stranded breaks in the DNA ahead of the fork. It lets the supercoiled DNA swivel around the break to release the tension, and then it instantly reseals the cut. It's like a molecular stress relief valve.
Incredible. Okay, so the fork is open, stabilized, and the tension is managed. Now, synthesis. You mentioned the golden rule. DNA polymerase only works in the 5' to 3' direction.
Always. And that's fine for one strand, the leading strand. Its template runs 3 to 5 fit relative to the fork's movement, so the new strand can be synthesized continuously, 5 to 3 minutes, just chasing the helicase as the fork opens. One long, smooth piece.
But the other strand, the lagging strand, its template runs the other way, 5 to 3 foot relative to the fork. So synthesis has to go backwards, away from the fork.
Exactly. It has to be synthesized discontinuously. Primus has to jump ahead, make a new RNA primer. Then DNA polymerase synthesizes a short stretch backwards, away from the fork until it hits the previous fragment. These short, discontinuous pieces are the famous Okazaki fragments.
Right, named after Reiji Okazaki. So the lagging strand is synthesized in these little backward jumps.
Little backward jumps, each one needing its own primer. Now, in E. coli, the main workhorse polymerase for both leading and lagging strands is DNA pol 3. It's incredibly fast and highly processive, meaning it can add many nucleotides before falling off.
And it has proofreading, right, to catch mistakes.
Yes, crucial. It has 3 to 5 foot exonucleus activity. If it accidentally adds the wrong nucleotide, it can pause, back up one step, snip out the wrong base, and try again. That's key to achieving that incredible fidelity, that less than 1 in a billion error rate.
Okay, so pol 3 builds the bulk of the DNA. But on the lagging strand, it eventually runs into the RNA primer of the Okazaki fragment made just before it. It can't remove RNA, can it?
No, pol 3 stops there. That's when you have a personnel change. Pol 3 dissociates, and DNA pol head comes in.
The cleanup crew.
Pretty much. Poly has a unique ability, 5 to 3 month exonucleus activity. It can chew away the RNA primer nucleotides ahead of it while simultaneously using its polymerase activity to fill the gap behind it with DNA nucleotides. It's like editing and replacing in one go.
Ah, okay, so pol the third for speed and bulk synthesis, pol primer removal and gap filling. Very neat division of labor.
And once pol first has replaced all the RNA with DNA, there's still one tiny nick left, a break in the sugar phosphate backbone between the fragment pol I just finished and the fragment ahead.
The final seal.
That's the job of DNA, Elise. It uses ATP to create that final phosphodiester bond, sealing the nick and joining the Okazaki fragments into one continuous, complete DNA strand. It's this beautiful, coordinated dance of enzymes.
That bacterial system is intricate but elegant for a relatively small circular chromosome. But when we scale up to eukaryotes like us, with our massive linear chromosomes packaged into chromatin, things must get way more complicated.
Oh, massively so. The fundamental chemistry is the same, 5 foot to 3 foot, synthesis, primers, polymerases. But the scale and organization demand a whole new level of control and additional machinery.
First off, size. Our genomes are huge. You can't just start at one origin and hope to finish in time, right?
Absolutely not.
Yeah.
Eukaryotic chromosomes have multiple origins of replication, often thousands scattered along each chromosome. Replication starts at many points simultaneously and proceeds bidirectionally from each, creating these replication bubbles that eventually merge. Think of mice. The sources mention they have something like 25,000 origins.
25,000. Okay, but that raises a huge control issue. How does the cell make absolutely sure that every single bit of the genome is copied exactly once and only once before the cell divides? You can't have some bits copied twice and others missed.
That's where replication licensing comes in. It's a brilliant control mechanism tied tightly to the cell cycle. During the G1 phase, before DNA synthesis begins, special proteins called replication licensing factors bind to all the origins. This essentially licenses them, marking them as ready to replicate.
So only licensed origins can start replication when S phase begins.
Exactly. And crucially, as soon as an origin initiates replication, its license is destroyed or removed. And new licenses cannot be loaded onto origins until the next G1 phase, after cell division is complete.
It's like a one-time ticket for replication per cell cycle. Get your ticket punched in G1, use it in S, and you can't get another one until the next cycle starts.
That's a perfect analogy. It ensures the entire genome gets copied once, and only once. It's critical for preventing gene amplification or loss.
Are the enzymes also different, like the helicase?
The core functions are similar, but the specific proteins are different, and there are some mechanistic distinctions. For instance, the main eukaryotic helicase, part of the MCM complex, actually gets loaded onto the origin in G1 onto double-stranded DNA, along with the licensing factors. It's activated later in S phase. This differs from the bacterial helicase that typically binds to already unwound single strands.
And the polymerases. Instead of just pol 1 and pol 3 doing most things,
Eukaryotes have a whole family of DNA polymerases, like 15 or more, with specialized roles. But for the main replication job, 3 are key. Pol, alpha, works together with primus to make the RNA primer and then adds the first stretch of DNA. Then it hands off to pole epsilon for continuous synthesis on the leading strand, and pole delta for the discontinuous synthesis of Okazaki fragments on the lagging strand.
So a more specialized division of labor compared to bacteria.
Definitely. Pole epsilon and delta are the high-fidelity processive workhorses analogous to pole the third, while pole alpha is more about initiation.
But beyond the sequence itself, eukaryotes have that other massive challenge. Our DNA isn't naked in the cell. It's wrapped around histone proteins forming nucleosomes, the basic unit of chromatin. What happens to them during replication?
The nucleosome problem. That's a huge logistical hurdle. Think about it. The replication machinery needs access to the DNA template, so those nucleosomes have to be temporarily disassembled or moved out of the way just ahead of the replication fork.
Okay, you unpack it.
But then, immediately behind the fork, as you now have two double helices where there was one, you need to reassemble nucleosomes onto both daughter DNA molecules. You need to essentially double the chromatin structure along with the DNA sequence.
That sounds incredibly complex. Where do the new histones come from? Are the old ones reused?
It's a mix. The cell synthesizes large amounts of new histone proteins during S phase. As the fork passes, the old histones from the parent DNA seem to be distributed somewhat randomly to the two new daughter duplexes. And then new histones are added to fill in the gaps, restoring the proper nucleosome density.
And this has to happen quickly and accurately.
Very quickly. It involves specialized proteins called histone chaperones and chromatin assembly factors that guide the process, ensuring the histones get loaded correctly. And importantly, they also help reestablish the epigenetic marks on the histones, which are crucial for gene regulation, onto the new chromatin. It's not just copying the DNA, it's copying the structural information encoded in the chromatin too.
So it's replicating the genetic code and it's packaging instructions simultaneously.
You got it. It's a whole other layer of replication complexity.
Okay, all this intricate machinery for linear chromosomes, it leads us to what you call the existential crisis, the end replication problem.
Yes. This is a direct consequence of having linear DNA ends and the fact that DNA polymerase needs that RNA primer.
Right. Walk us through it again. Why is the end a problem?
Okay, think about the lagging strand again. Synthesis happens in Okazaki fragments, each starting with an RNA primer. DNA polymerase removes those primers and fills the gaps. Fine. But what about the very last primer, right at the extreme 5-foot end, the newly synthesized lagging strand? When that final RNA primer is removed, there's no upstream Okazaki fragment. There's no existing 3-OH group for DNA polymerase, like POL1, to extend from to fill that gap.
So removing the last primer leaves a gap that cannot be filled by standard DNA polymerase activity.
Exactly. You end up with a short, single-stranded overhang on the template strand. And the newly synthesized lagging strand is shorter than its template. With every round of cell division, this process repeats, and the chromosome gets progressively shorter from that end.
And if that shortening eats into actual genes, that would be catastrophic.
It would eventually lead to loss of essential genetic information. That's why eukaryotic chromosomes have protective caps called telomeres.
Right, the repetitive sequences at the ends.
Yes, in humans, it's typically this repeating sequence TTAG over and over for thousands of base pairs. These telomeres don't encode any proteins. Their job is basically to act as a disposable buffer.
So the chromosome can afford to lose a bit of telomere sequence each division without losing vital genes.
Precisely. And they serve another critical function. They prevent the cell's DNA repair machinery from mistaking the natural chromosome end for a dangerous double-strand break, which could lead to disastrous chromosome fusions.
Okay, so telomeres are the buffer, but they still shorten. Eventually, wouldn't they run out? The sources link critically short telomeres to cell aging senescence, right?
They do. In most of our somatic cells, regular body cells, the telomeres do shorten over our lifetime. When they reach a critical length, it triggers a signal for the cell to stop dividing or even undergo programmed cell death, apoptosis. This is thought to be a tumor-suppressing mechanism, limiting the number of times a cell can divide.
But some cells need to divide indefinitely, like stem cells or germ cells that make sperm and eggs. How do they cope?
They have the crisis solver, an enzyme called telomerase.
Okay, what does telomerase do?
Telomerase is amazing. It's a ribonucleoprotein, meaning it's made of both protein and RNA. And crucially, it carries its own internal RNA template within the enzyme itself.
Wait, hold on. It brings its own template to the chromosome end?
Yes. That RNA template base pairs with a single-stranded 3' overhang left on the chromosome end, the G-rich strand. Then, using that RNA as a guide, the protein part of telomerase, which is a reverse transcriptase, synthesizes new DNA repeats onto the 3' end of the chromosome, effectively extending it.
So it directly adds back the lost telomere sequences, counteracting the shortening?
Exactly. It extends the longer 3' ending strand. Then, the regular replication machinery, polymerase, can come in and synthesize the complementary strand, fully restoring the telomere length.
That's wow. So telomerase activity is high in cells that need to divide a lot, like germ cells and stem cells, keeping their telomeres long.
Correct. But in most of our differentiated somatic cells, telomerase activity is very low or absent, which leads to the gradual telomere shortening associated with aging.
And this connects directly to those premature aging diseases mentioned in the sources, like progeria or Werner syndrome. They have defects related to telomere maintenance.
Yes, they often involve mutations that lead to abnormally short telomeres, accelerating the aging process at a cellular level.
But there's a dark side to telomerase, isn't there? The cancer connection.
Absolutely. While silencing telomerase in most body cells helps prevent uncontrolled division, cancer cells often find ways to reactivate it. The sources state something like 85% or more of human cancers show high telomerase activity.
Which basically gives them cellular immortality. They overcome the normal limit on cell divisions imposed by telomere shortening.
Precisely. They can keep dividing indefinitely, which is a hallmark of cancer. So telomerase is this double-edged sword,
Yeah.
essential for germline continuity in stem cells, but also a key enabler for cancer when reactivated inappropriately. #outro.
Wow. Okay, so stepping back from this deep dive, the contrast between the streamlined circular replication in bacteria and the incredibly complex, regulated, multi-stage process in eukaryotes is stark.
It really is. From needing just one origin versus thousands, the ddNA initiator versus that whole ORC and licensing factor system in eukaryotes, the way helicase binds, managing the nucleosomes.
And of course, the whole end replication problem and the telomerase solution, which bacteria just don't need. It really highlights the evolutionary adaptations driven by genome size and structure.
It's a system that had to evolve incredible layers of control to handle the complexity of eukaryotic life.
The speed, the accuracy, the packaging, it's all remarkable. But that telomerase aspect, the trade-off it represents, feels particularly profound.
It really does. As we've discussed, you need telomerase for continuously dividing cells, for organismal development and longevity in a sense, but its absence seems linked to aging, while its presence is critical for most cancers.
Which leads us to a final thought for you, the listener, to mull over. Given this tight connection essential for some cells, dangerous in others, linked to both aging and cancer, what does the incredibly strict biological regulation of telomerase activity tell us about the fundamental balancing act life performs? That trade-off between, say, unlimited regenerative potential and the risk of uncontrolled growth.
And thinking ahead, what might be the implications if we could learn to safely modulate telomerase activity? Could activating it carefully extend health span, or could suppressing it more effectively offer new avenues for cancer therapy? It's a fascinating area right at the intersection of aging, disease, and the very mechanics of copying life's blueprint.